Today topic is the structural lipids in the membrane. Venous lipids की हम बात करेंगे कि what are the role of the venous lipid और वो किस तरह के होते हैं, कितने टाइप्स के होते हैं और क्या क्या फंक्शन हैं. Most of these lipids contain the polar head group and the non-polar tail, usually attached fatty acids. Diversifications can come from modifying a different backbone and changing the fatty acids, modifying the head group. So these are the three major reasons which can change the properties of which can make the diverse the structural lipids. The properties of the head groups determine the surface properties of the membrane. Different organisms have different membrane lipids head group compositions. This is not necessary that all the organisms have the same type of head group in all the organisms. Different organisms have different head groups for their lipid in the membrane, lipid moiety in the membrane. Different tissues have different membrane lipid head group composition. Uh, as for the different tissues are conformed, same procedure can happen over there. The first type of lipids are the glycerophospholipids. These are the primary constituents of the cell membrane. Two fatty acids form the ester linkage with the first and the second hydroxyl group of the glycerol 3 phosphate. Head group is charged at the physiological pH. Glycerophospholipids, these are the derivatives of the phosphatidic acids. These are also known as the phosphoglycerides. These are the membrane lipids. Fatty acids, mostly in this um, in these kind of lipids, there can be two fatty acids attached with the glycerol molecule, and these two fatty acids are mostly ester linked at first and the second carbon, and on the third position of the glycerol, there may be a phosphodiester. There may be a polar charge group with the polar charge group is attached with the phosphodiester linkage. Okay, just need to explain. Just a minute. I'm looking for the pointer. This is the glycerol molecule. This one is the glycerol molecule. And with this glycerol molecule, the position number 1 and position number 2, mostly the fatty acids are attached over there. Here the fatty acids are mostly attached. This is actually the glycerol 3 phosphate. This is actually the glycerol 3 phosphate. There are uh, um, three carbon in this molecule. There are three carbon in this molecule. And on these three carbon number 1 and carbon number 2, uh, the fatty acids are mostly attached. Fatty acids are mostly attached in these two positions. At these two positions. Here is a question for this molecule, that this molecule contains the three carbon. ये molecule तीन carbon है इसके अंदर. अब ये कैसे डिसाइड हो कि ये ग्लिसरॉल 3 फास्फेट है, ये ग्लिसरॉल 1 फास्फेट है. फॉर एग्जांपल अगर हम नंबरिंग यहाँ से स्टार्ट कर दें और इसको देखें तो इसके हिसाब से तो इसको ग्लिसरॉल 3 फास्फेट के बजाय ग्लिसरॉल 1 फास्फेट होना चाहिए, बट दिस नंबरिंग इज according to the stereochemistry. This is actually stereo numbering. So इस वजह से मोस्टली इसके साथ SN ग्लिसरॉल 3 फास्फेट लिखा जाता है, यानी कि इसको स्टीरियो नंबरिंग दी गई है. अकॉर्डिंग टू द स्टीरियो केमिस्ट्री हम इस मॉलिक्यूल को लिखेंगे और इसका जो सेंट्रल मॉलिक्यूल है, इसका जो सेंट्रल कार्बन है, दैट सेंट्रल कार्बन विल बी कार्बन नंबर 2. और इस कार्बन नंबर 2 के ऊपर वाला कार्बन कार्बन नंबर 1 होगा और इसके नीचे वाला कार्बन कार्बन नंबर 3 होगा. तो इस हिसाब से जो नीचे वाला कार्बन है, कार्बन दिस इज कार्बन नंबर 3 और फास्फेट ग्रुप इज अटैच्ड विद दिस कार्बन. So this molecule is glycerol 3 phosphate. If we need to write the glycerol 1 phosphate, we have to attach this phosphate group with the carbon number 1. So this molecule will be carbon number then this molecule will be glycerol 1 phosphate if the phosphate group is attached here. at this position. So, mostly the phosphate group is attached with the carbon number 3. So this is the glycerol 3 phosphate. Unsaturated fatty acids are commonly found connected to the carbon number 2. The highly polar phosphate group may be further esterified by an alcohol. Such subsequent groups are called the head group. You can't understand. I'm saying that, say for example, this is glycerol molecule. This is glycerol molecule. Uh, Here the fatty acid is attached. Say for example, here the fatty acid is attached, and this is the carbon number 3. This is esterified by the phosphate group. This is esterified with the phosphate group. So this is these two fatty acids are attached with this polar group. Okay. This phosphate group can further be esterified with another alcoholic group. So for example, with the X. Okay. This is further esterified with some other alcoholic group. So this complete molecule will make the uh, head head mostly at these position. Uh, Phosphatidylcholine, you can say choline or the ethanolamine are attached. Ethanolamines are attached. So we can say mostly at these position, phosphatidylcholine or phosphatidylethanol may be present. Both of these are present and linked with this head group through the phosphodiester linkage. And at the neutral pH, they may have a negative charge over there. There are the negative charge because these are the membranous lipids, so negative charge is important to interact with the environment. These are the examples of the glycerophospholipids. Name of the glycerophospholipids. Maybe there may be phosphatidic acid may be present. Phosphatidylethanol, ethanol amine is present. Phosphatidylcholine, choline may be present. Phosphatidylserine, serine group may be present. Sometimes one more glycerol molecule is attached, so phosphatidylglycerol may be present, or sometimes in acetol or phosphatidylglycerol may be present over there at the position of the alcoholic group to make the head of this molecule. Phosphatidylcholine is the major component of the most eukaryotic cell membrane, and many prokaryotes, including the E. coli, cannot synthesize this lipid. Their membranes do not contain the phosphatidylcholine molecule. So this is mostly present in the eukaryotic cell membranes. Next are the ether lipids, or we can say plasmalogens. Vinyl ethyl analog of the phosphatidylethanol and this is common in invertebrate heart tissues, also found in some protozoa and in aerobic bacteria. They perform these are important to work to resist to provide the resistance to cleavage by common lipases, but cleaved by the few specific lipases. What is its proper function? It may be involved in increasing membrane rigidity. It may be involved in the source of signaling lipids. It may be involved in the and antioxidants. This is the structure of the plasmalogen, where the ethanol amine is attached. In such kind of lipids, at one position there may be instead of the ether linkage, there may be an ester linkage, and there may be instead of ester linkage there may be an ether linkage. Ether lipids में at one position ether linkage होती है, दूसरी position पे ester linkage होगी, लेकिन एक position पे ether linkage होती है. Like say for example plasmalogen, here you can see that ether linked alkane are here. This one is the ether link. Lipids is ether-linked phosphoglycerides may be present in the platelets activating factor. Aliphatic ether analog of the phosphatidylcholine. Acetic acid has esterified deposition number 2, carbon 2. Acetic acid may be attached over there. First signaling lipid to be identified. This is the signaling lipid. As you can see in this diagram, this is the ether-linked alkane, and here the acetyl ester is present. It stimulates the aggregation of the blood platelets and play a role in the mediation of the inflammation. This is very important to important factor for activating the blood platelets. The next molecule is the sphingolipids, and we will try our best to um, mm, discuss this topic by tomorrow. Thanks a lot for watching this. Good luck, and I hope you will be understanding. It will be easy for you to understand. Thank you. Thanks a lot.